Let's go over somatic hypermutation for the MCAT. Welcome back to day 10 of MCAT 528 and 28. Starting off, we have immature B cells, which form in the bone marrow. They then migrate to the lymph node. The now mature B cells get activated by helper T cells, pathogens, or follicular dendritic cells. Here, intracellular enzymes then introduce point mutations to the variable region, also known as the antigen binding site of the antibody DNA. This process is known as somatic hypermutation, and it could produce daughter cells that either have decreased or increased affinity for the antigen. The disadvantaged mutations undergo apoptosis, while the ones with higher affinities undergo class switching and differentiation, which produce antibodies that enhance phagocytosis, neutralize pathogens, and activate the complement system, or helper B cells. Affinity maturation, where the daughter cells of the mature B cells, which have higher affinities for their antigen, are able to proliferate further. This process is also known as clonal selection and clonal expansion. You should know that clonal selection is the process where the daughter cell with the higher affinity is selected, whereas clonal expansion is where that specific cell will rapidly multiply in order to fight the disease, which occurs through it outcompeting the other daughter cells that have a lower affinity for the antigen. Now, in biology, we usually look at mutations as bad things, but why are they so important in the immune system? You should think of it like a predator and prey arms race. As viruses constantly mutate, as well as new pathogens are introduced to our body, we need more and more antibodies to be able to protect us from the variety of different antigens.